hope this isn't too disappointing for you. Some of the things that you'll hear about today may not be some of the things that you expected to hear about DNA in the Book of Mormon. But the purpose of this particular paper this morning is hopefully to inform and then to educate you on genetic matters so that you will be able to make some judgments concerning the genetic data that has come out and the genetic data that most certainly will come out concerning Native American studies. Recently, and not so recently actually, there have been a number of individuals who have suggested that there is a simple way to determine the validity of the Book of Mormon by analyzing and comparing Hebrew and Native American DNA. Right off the bat, I have a serious problem with that particular statement and with this hypothesis. I have a question as to whether DNA is an adequate system at all to determine the validity of the Book of Mormon. The questions that genetics are designed to answer are things about genes and the distribution of genes in populations. And I'm going to talk a little bit about that as we go along. But what may seem to some as a simple test can actually be quite complicated. Population genetics studies involve a number of inherent difficulties. This paper is an attempt to outline some of the issues involved in human population DNA studies in order to demonstrate, I hope, the difficulties in making determinations such as those that have been suggested. What I would like to do is first bring you up to speed on some of the different types of DNA analysis that we deal with. The one that most of you have heard most of, because that's what's been done the most of, is mitochondrial DNA analysis. Mitochondrial DNA comes from a very small molecule that sits inside of our cells. It's a molecule of about 16,500 base pairs in length. It's a small circular DNA molecule. It only contains about 1 to 200,000 of your genetic information. It's a very, very, very small contribution of your total genetics. But it has been studied quite extensively, particularly in populations, primarily because of the ease at which we can study mitochondrial DNA. One of the reasons is that it's so small, and that we know a lot about the specific DNA sequences that are on the mitochondrial DNA. It also has a very specific type of inheritance called maternal inheritance that I'll go into in just a minute. And it has a technical property that called non-recombining, in that it doesn't get mixed up from generation to generation, as we'll see as some of the other genetic systems that we're dealing with do. And another very important part is this term that's called linkage disequilibrium. What it means is that all of the markers, all of the different parts of a single mitochondrial DNA are inherited together and intact, which means that it doesn't really matter too much how many different markers you look at on a piece of mitochondrial DNA. If you look at one, it's going to be linked to a second one, and it's going to stay linked to that for many, many generations down through a population's history. And so we are essentially looking at a single genetic locus when we look at, or a single genetic piece of information when we look at mitochondrial DNA, as opposed to the rest of the genetic information that we have that comes inherited equally from father and mother that may have hundreds of thousands of different loci. And that's important for you to remember as we go along. There's another type of inheritance that we'll talk about that has also received a fair amount of prominence in Native American studies, and that has to do with the Y chromosome. The Y chromosome is a piece of DNA that is inherited from father to son and carries the male determining factors. So this is the chromosome, this is the piece of DNA that carries the genes that are responsible for making males males. Okay? So the females don't have it. So you can see there will be a difference in the genetic inheritance patterns of mitochondrial DNA, of which all of us have, except that we all received it from our mother, and Y chromosomal DNA, which only males have, who received it from their father. There's also some of the same situations associated with mitochondrial DNA. It has a very specific type of inheritance. It's a small piece of DNA. The genes on it, contrary to what most males would like to believe, don't do a whole lot. All they do is make us male. And the reason we know that is because females get along just fine without that chromosome, right? They carry a lot of other baggage, so we won't talk about that. So paternal inheritance, but with the Y chromosome, but the bulk of our genetic information, and that gives us the most knowledge about who we are and how we're related to individual families, extended families, tribal groups, populations, and the world, happens in our autosomal DNA. Though that DNA that's carried in our chromosomes that we receive 50% from mom and 50% from dad. That's essential. And that's where the bulk, by far, of the genetic information is contained. It comes equally for both males and females. There are thousands, tens of thousands of independent loci, or independent markers that we can use to follow inheritance from generation to generation, as opposed to, say, mitochondrial DNA, which have just a handful, and those handful are completely linked in sets. Could you hit that P for me up there? I went too far. Okay. Mitochondrial inheritance, remember, comes down from the mother to her children. It comes down both to males and females, but it's solely inherited from the mother. And so if we look at individual number 2, who is the mother up here, gave her mitochondrial DNA to individuals 4, 5, and 6, who gave it to 10, who gave it to 15, who gave it to 17. So you can see the inheritance of mitochondrial DNA. My mother's mitochondrial DNA has ended up like number 9 over there, because my mother didn't have any daughters. And so her mitochondrial DNA, as far as she's concerned, has come to an evolutionary dead end. It's stopped. It hasn't been transmitted to the next generation. And it is the DNA of my wife and my brother's wives that have contributed to my children's mitochondrial DNA. So you can see that the mitochondrial DNA that I have, that I inherited from my mother, has not been transmitted to the next generation through me. And so there are some real complications in following mitochondrial inheritance in populations. We'll come back to that. Y chromosome, a lot of the similar types of things, except we're only dealing with males at this time. So father to son to son to son to son. Females are completely left out on Y chromosome inheritance. Parents would change positions with each other, and in reality, when we look at the black one or the red one down here in the children generation, we would see a combination of red and black for those chromosomes, and that's how it really happens. We do have a pretty good handle on how that recombination happens, and we can deal with that in using that to reconstruct population genetics. If we limit ourselves just to identification of populations by either the mitochondrial DNA or by the Y chromosome DNA, we're leaving out the bulk of the ancestral information. If you see a traditional genealogy chart as you would construct it, often we have the males on the top and the females going down the bottom. You can see the Y chromosome line going across the top. And we can identify the males in that line based on your Y chromosome because it will all be the same. Likewise, we can identify all of the females on the mitochondrial line that comes down across the bottom because they will all be the same.